to then also study the process of organic matter of contribution to carbon in the soils. And more recently, we are experimenting with producing mycorrhizal labeled tissue to do research about mycorrhizal contribution to carbon. But we can also do experiment within the chambers, for example, in a collaboration with Courtney and C. Fonte, where we are doing actual research inside the chamber. What we then do with the plant that we produce is typically we use the mixing model to determine how much of the carbon and nitrogen from the plant material either contribute to the bulk soil or a soil physical fraction. We tend to use a physical separation of organic matter. We have traced that in gases and also in specific compounds like PLFAs and amino sugar, mostly as microbial biomarker. And we are also trying to do some nanosims if we have enough of the enrichment with collaboration, in particularly with Germany. The first experiment that we did in the field, also with as part of Jenny Sung's dissertation, it was at the Konza Prairie, where we incubated that litter for 3 years. And actually, we were very excited because we were there just last month to pick up the 10-year sample. And so we'll be happy to see what happened after 10 years. But what that shows is that when you don't have a litter bag that confines the litter, all the litter goes down to basically zero mass. But in the process, it forms some carbon, but not with the same dynamic of the mass loss. We have some early formation and some later formation, and not much in between. And when we looked at this data together with the microbial biomarker and the physical fractions, we came to propose what we call the 2 pathways of soil organic matter formation, where basically the idea is that the soluble that get produced, in particularly in the early phase of decomposition, when all the solubles that were originally in the litter material get leached, but also a little bit throughout decomposition as the microbe break some of the structure that becomes soluble and enter the soil as dissolved organic matter. And those are assimilated pretty efficiently by the microbes. And then we found them in the mineral fractions. On the other hand, the more structural component, particularly rich in lignin, they get fragmented, and we also, with the collaboration with Diana Wall, we followed the effect of the fauna and looked at their role in actually helping with that fragmentation and the formation of what we call the particulate organic matter in soil. These 2 pathways of soil organic matter formation is actually pretty contrasting with what was the prevailing idea that organic matter form as a continuum, mostly with the attention to the particulate again, and not thinking about the soluble contribution from the litter. But thinking that plant litter residues basically are continuously processed by the microbes, the microbial contribution to them becomes more and more, and as they get smaller, they then associate to the organic matter. And so our proposal was, our hypothesis was different from the prevailing idea, and so it required a lot of experiment to test it, and that's what we have done in many different ways, and I'm only going to show you a few, but we consistently actually got confirmation of our 2 pathway model. One of the hypotheses that I like the most that Michelle Addix did in the lab, because maybe I define that as my most geeky experiment, where what we did was we differentially labeled, both with 13C and 15N, the metabolic fraction of the plant material from from the structural fraction so that we could follow both of them, applying a triple model, both to the contribution to the respiration, but also to the contribution of the fraction in the soil. And what we observed was that basically the mineral-associated organic matter was only formed by the soluble component, what we call the metabolic component. Basically, it's the hot water extractable. Whereas the structural component, despite they were used by the microbes, they actually only formed the POM in the soil. And we are finding confirmation of that over and over. Now we have an experiment ongoing at RDEC where we incubated exudates and root structures from 2 different corn varieties. And the exudates are actually mostly contributing to the mineral-associated organic matter. Those 2 columns are the 2 varieties, whereas the root structure are mostly contributing to the POM. And so that is another confirmation of the 2 pathway. But what was particularly interesting, because it also allowed us to follow it over time, is an experiment that we did with Michelle in collaboration with Ed Gregorich in Canada, where he had incubated 13C labeled residues in 10 different sites along a texture gradient in Canada, as well as a climatic gradient. And what we find out was that after 5 months, the first column at each site, the majority of the carbon from the residue was found in the free particulate organic matter, but already you had the mineral-associated organic matter, again pointing to the dissolved organic matter contributing to it, and not necessarily derived from the decomposition of POM. But that was further evidentiated by our 5-year sample, where basically the free POM was entirely lost at almost all the sites, whereas the MEOM was basically maintained and didn't increase much, suggesting that the decomposition of the POM didn't actually contribute to more MEOM formation. What was interesting is that the POM protected in the aggregates remained a bit, again suggesting another thing that we know and we are studying further is that aggregates can protect POM from faster decomposition in the soil. And so that's again was a confirmation of our 2 pathway hypothesis of the independence of POM and MEOM. And so if today I had to rewrite the MEMS paper, I would rewrite it thinking not just of the mineral-associated organic matter, but also of the particulate organic matter, which is a very important component of soil. And that's more contributed from the structural component of plant material, as well as from some microbial structures. And so this has now become really the framework that we are using in the lab. And Jocelyn Lavalley is working a lot with me with defining it further and further. And the idea here is that in soil, if you want to understand how soil works, we really need to separate these 2 contrasting component, the particulate organic matter, which is mostly formed of plant residues and microbial structural residues, from the mineral-associated organic matter that is produced more from soluble material and microbial necromass. And the mineral-associated has the caveat to requiring more nitrogen because it's contributed more from microbial component, but on the other hand, is protected by the mineral association and therefore has a slower turnover on average. There is also some faster turnover mineral-associated organic matter. But we are now stressing this framework because we believe that POM and MEOM have actually contrasting vulnerability, potential to sequester carbon, and respond differently to management. While those fractions make sense conceptually, from a quantification standpoint, it ends up to be procedurally defined, and they can be defined by size or density. And there are a lot of different ways in which they can be separated. And so in a way, the picture, the scientific picture is by far more complex than just a POM and a MEOM. And we are for sure digging down into it. But I still want to point out that the POM and MEOM framework are very useful to move ahead and provide solution. And but if we want to do that, we need to be able to measure them at scale. And so that's another area that we are pointing in our lab, is trying to see how can we increase the throughput of those analyses that, if any of you has ever done them, are actually very time consuming. And so on one hand, we are working with Seed Zero here, CS, for Collins, to try and engineer and automatize the operation so that we can have an instrument that does the density separation. That is the most tedious step in the fractionation. Another thing that we are doing in collaboration with Francisco Calderon and Paulina Ramirez is to try and understand if we can use FTIR to actually predict those fractions. And that seems to be having some very interesting results. But also another approach that we do when we fractionate large data sets that have a lot of other data is to use artificial intelligence technique to try and predict those fractions, basically just fractionating a subsample of the soil and then using artificial intelligence to project them at large scale. This is a work that I've done during the sabbatical in Europe, where being at the Joint Research Center of the European Union, I came across the Lucas data sets. There was an impressive data set with over 20,000 samples taken all across Europe in many different sites. And what we did was to subsample that data, set of soils, and extract 400 samples that represented the range of texture as well as the range of organic carbon in the soil. And then using artificial intelligence, we basically projected the measured fraction all across Europe and produced maps of how the carbon, in particular organic matter or in mineral associated organic matter, is distributed across Europe. And what came of great interest is that actually the mineral associated organic matter that we always think of as the most persistent fraction, it is the most persistent fraction, and that's why it's actually most abundant in the soils that have low organic carbon. And so that's kind of counterintuitive, that the soils that in Europe have been mostly used for agriculture are now the ones that have the highest amount of MEOM, while they have the lowest total carbon. And in fact, if we break down this result into the different land uses, what we saw is that in Europe, the conifer forests in Scandinavian region in particular are the ones that store the most amount of carbon when quantified on a megagram of carbon per hectares. And they have the majority of it, in particular the organic matter, with a relatively high carbon to nitrogen ratio. On the contrary, arable are the ones that have the lowest amount of carbon, but the majority of it is in mineral associated organic matter, again showing how that is the most persistent fraction, the one that persists disturbance as well. But you can also notice the different carbon to nitrogen ratio across the the different land uses. Again, in particular, because of the crop we grow, but also because of the high fertilization in arable land. So nitrogen is actually not necessarily an issue, as others say, about carbon sequestration, because in particular in arable soils, there is a ton of nitrogen that needs actually to be used to sequester more carbon. And that's evident when we look at the total carbon in Europe. So we multiply it by the land area, and we see that arable actually are the second largest system for carbon in Europe. And so if we want to, this information is very helpful if we want to target specific management practices, because we know which, for example, that we need to protect the carbon in this forest, or how can we manage arable to stock more carbon, in particular to increase the palm in arable soil. The other things that we learned from this database is that the mayom saturate. So this was a theory that actually Cathy Stewart, Johan Six had proposed, but we first demonstrated, we're the first demonstration at very large scale. And so when we think about carbon sequestration, we need to realize that the mayom saturate while the palm never saturate. On the other hand, right now, the majority of the soils are far below saturation. And so we shouldn't be worried about saturating the minerals when we think about carbon sequestration. There is a lot of potential to sequester carbon in soils. And those soils are only the 0 to 20 centimeters. And so if you go at depth, there is even more opportunity for carbon sequestration. But I want to point out the importance of separating mayom and palm if we want to understand those dynamics and again inform solution. Another reason why we do need to separate mayom and palm is because they have a very contrasting response to global change. And so a lot of the time we don't have very clear, for example, in terms of elevated carbon dioxide effect on organic matter, the size of the pool doesn't change. But what Kathy Rossi in her PhD study with a meta-analysis observed was that, yes, for example, elevated CO2 doesn't change the total carbon stock, but very much change the distribution. And the same is for warming, whereas nitrogen fertilization doesn't change the distribution, but increase the size. And we have an interesting hypothesis for how these different drivers can affect independently the palm and the mayom to have those responses. And what I most I like to stress is the response with warming that is now, very much on our head. If the planet warm, soils will turn over faster and lose more carbon. But actually, what Katie found in this meta-analysis is that the POC is what is sensitive to warming, whereas the milk, because it's minerally protected, is actually much less sensitive. And so separating those fractions can really help us disentangling the effect of climate change and other changes on soil carbon. But when we do this manipulation experiment, we also need to be careful with how do we manipulate the system. And for example, what Katie analysis showed is that warming experiment that only warm the air basically do very little to the soil, whereas warming experiment that warm the soils have a much dramatic impact. And so again, it's very important that we do the right thing when we want to study soil responses to global changes. And all this understanding, it's great, of course, to advance our science, but I also felt to a point that we really needed to move this understanding into tools in order to both have tools that can help us refining our scientific inquiry, but also tools that can be used for decision making. And that was the driver to actually get into modeling or collaborate with modelers to try and have a modeling tool that better represent this advanced understanding of how soil organic matter turnover and persist. And we started off with a project with Nell Campbell, where we worked at what we call the Lidl model, also with Bill Parton and others, where Nell basically modeled the litter decomposition component that I told you about, and the idea that not only the structural litter component, but also the dissolved organic matter entered the soil, and that the microbe and microbial efficiency played an important role in the process. Then Andy Robertson joined my group as a postdoc, and he took the Lidl model and built it, what we call now the MEMS 1.0, because what it did was to then link the soil component to the litter and have all the different soil organic matter fractions. And the beauty, in my opinion, of this model is that all the pools are measurable and all the fluxes are biophysically defined, and some of them also measurable. And so that offers an opportunity for a very thorough verification of model output. And for the MEMS 1.0, we use those Lucas data at the European continental level, and we got pretty good results. Also, if you look at the residual, in few cases we had either low or high, but overall the map is green, saying that we were doing pretty well with this model. However, this was just a soil carbon model. And of course, all of you know that if we want to project carbon responses, we have to link them to nitrogen, we have to link them to plant microbes and so forth. And so Yao Zhang and Jocelyn Lavallee took over from the MEMS 1 and actually built what we call the MEMS 2.0 model, which is a full ecosystem model. We are very excited to, I think it's one of the manuscripts on my desk that I hope to get to very soon and send out for publication. And again, the beauty of this model is that everything is measurable. It includes the two pathway model, also include work from Noah Sokol and Mark Bradford, where we separated the below ground input from the above ground input, include exudation and a lot of other process, and also explicitly have different layers so that you can model the carbon and nitrogen dynamics to that. We are really excited about this tool, and so are a lot of our a lot of others, and so we have a lot of requests for collaboration and developing it further. But to start with, we calibrated and validated it in grasslands. And Rebecca Evan in my lab, she has fractionated all the neon soil. So in all the different the what I call the neon megapit soils all the way to depth. And then Yao used those measured data to, on one hand, calibrate across 4 different grasslands in the neon network and then validated across other 2 grasslands. And as you can see, of course, it's not perfect, but we are doing pretty well. And that's the first iteration of the model. And the other nice thing about this model, it's that it can be validated also with flux data. And so in systems that have eddy covariance or measure ET, we can also validate the model at a shorter time scale or a continuous time scale with those data. And so again, now we have a lot of work to do with this model. We want to move into forest and arable land and represent different management practices. We also have a project to do MEMS tropics, and so it's one part that is really exciting of the research program. And finally, once we have all this framework and tool, the other part of the research that we are working on is the management solutions. And we are working on many different projects, a lot also in forest, but I selected the studies that are in one way most providing the most exciting results. One is the work that Sam Morger has just completed in the southeast of the United States. We studied 5 pairs of grasslands from Texas to Alabama and so forth in the southeast of the United States, where within the pair, one farmer was doing conventional grazing and the other farmer was doing what we call AMP, or adaptive multipath of grazing. So the cows are moved all the time and they don't really consume the grass all the way down. Sam has done an amazing amount of work analyzing all those soils along the soil profile. And here are the average, where you see that all the way down to depth, the AMP had higher carbon stocks, but in particularly what made the difference was the mineral associated organic matter. And we think that that was mostly driven by the higher nitrogen in those soils, despite they were the soils that were not fertilized. And we are now trying to put all this data together, and Jocelyn is working at a synthesis, and we'll see what are the driver of those changes. In cropland, Laura van der Poel is instead looking at how, by intensifying and diversifying cropping system in our region, where if you want, the control system is the wheat fallow. And the idea here is that if we do continuous grain rotation, you would already start improving the soils. But our hypothesis was that by adding a legume in the rotation, then you add the nitrogen that further stimulate microbial activity, increase the efficiency of microbial transformation of plant inputs, and therefore would result in a higher soil health, but also in more meal. And while Laura did find that the topsoils of the continuous plus legume plus P had higher meum, and she analyzed also a lot of soils, both across in-farm, but also experimental station that had this treatment. She actually didn't find striking results. And so the idea that the continuous rotation and the addition of the P in the rotation would totally transform and regenerate the soil is actually, we're not that certain about it. And that confirms with other studies that, for example, Alison King did to her PhD, where she also didn't find those striking results. And so now she came to work with me with a USDA postdoctoral fellowship to investigate the driver for this, for those responses, and why in some sites you see them and in other not. And so she convinced actually 15 long-term sites to sample the soils for her, and she's now receiving all those soils, and she on one hand will do an analysis of those soils to where each of those have long-term either conventional or sites where they intensified or perennialized the system, and see what she find out. But then she's also designing a very interesting mechanistic experiments to try and parse out the effect of the carbon use efficiency from the microbial community versus the soil texture and pH and mineralogy. So that's an exciting project that she started. But to be honest, what I have my most faith on for regenerating soil is what we call regenerative agriculture. And that's a project that, you know, for the one of you that drive to Kansas, understand my joke that we call Aaron our wheat Jesus. But anyway, what Aaron is doing in collaboration with General Mills is looking at a lot of different farms in Kansas that adopted the 6 principles of regenerative farming, and which a lot of them are also adopted in the cropping system, where you have cover crops, so the continuous input, the deep root inputs. But what I think will really make the transformation is the inclusion of the animals in the system. That might kickstart the nitrogen cycle, maybe more than just adding legumes. And in particularly, what we need to regenerate soil health and regenerate soils is having enough input so that you can have you can recreate particulate organic matter in agricultural soil that so far are very deprived of particulate organic matter, stimulate that turnover so that you can stimulate the nutrient recycling, but at the same time with these additional inputs both from the plant and the animals, the microbe could use it more efficiently, and while they turn over the organic matter and produce fertility, they can also leak down some necromass to form mineral associated organic matter. And so I think that we will use this framework to analyze now soils from all these different regenerative practices and see whether that's true or not, that the animals might make the difference in the system. And so while I'm very excited, and of course the largest part of my program is researching and teaching and mentoring graduate students, I also think, and actually even more after last night, that we need to go out and we need to talk our science, and we need to make sure that kids in school develop critical thinking and develop an appreciation for science, but also talk with the people, understand what are the questions of the farmers, and try to break that gap and to bring our understanding in the rural community and educate them to appreciate science, as well as understand from them what are the questions that we need to work on and examining. So I also love and I'm very excited that actually everybody in my group shared that that love for outreach and education. And so in conclusion, for sure, the most important things we need to do is to alter fossil fuel emission. There is no intention to say that sequestering carbon in soil can be the solution to climate change. But it's important that we also do that, because we not only have to stop emission, but we need to draw down carbon from the atmosphere if we want to stabilize the climate. And the good news is that by doing that, we also increase soil health and foster resilient agricultural system. In my group, we are now working on this framework of the POM and MEOM, and we believe that is a better way to conceptualize organic matter and move our understanding forward. And using that understanding, we have developed this tool. And again, I'm very excited about the idea of using it more and more and apply this approach and this tool to address management solution. In closing, I want for sure to acknowledge all the funding without which I wouldn't be able to do all this research. And what has been most exciting recently is that on top of the regular funding we get from NSF, USDA, DOE, I've started now working more with industry. I'm very excited to a new project with Steve Rosenzweig at General Mills and Andy Robertson and Chris Davis at Shell, and also our grasslands project with McDonald's, and the work with Emanuele Lugato that we are doing at the European Commission. All this work would not be possible without the amazing student that I had from the very beginning. And I want actually to acknowledge my Italian students, with whom I've worked for 15 years when I was there, and they surely have contributed to put the foundation to this research. The amazing student that I had over the year, and collaborator and research associate and postdocs here at CSU. And really, for a soil ecologist, you couldn't be at a better place. These are all the soil ecologists in the picture at the Soil Ecological Society. We were the biggest group, from Diana Wall and Matt and John Moore and so many others. Again, all this work, this Phil Einerson was my advisor, and the great collaboration with Kathy and Carolyn, and of course my family have always supported me in this research. So thank you very much for the attention, and I hope sometime we have also some good news from somewhere else. Thank you.
Woo-hoo. And clap.
So I'm not sure how do we want to do the question, but